but we will move on, push on, because thanks to a generous gift from George Abraham, who is here, he essentially set this up to have lectures. We plan about 6 a year, with a balance between outside speakers and speakers within our institution. I am delighted that we got one in right before COVID, and then have been postponing and postponing. But to me, we got to get back to normal, and I can't think of any better way to get back to normal than to start having our dean's lectures. And I'm particularly thrilled today to have an internal lecture with scientists who have really are changing the face of the world with our knowledge of aging and cancer. So with that, I think I'll turn it over to Dirk and let him do the introductions. But thanks everybody, both live. I know we're streaming this on Zoom and on Facebook, so but welcome.
Thank you, Mark. So yeah, it's really a pleasure to invite Vera Gorbunova as the first of the internal Dean's Lecture presenters. As Mark said, this series had been interrupted by COVID for a while, and we hope that we can kind of pick it up again. We have a number of speakers lined up, and we'll see whether that will happen. Some interesting external speakers, as well as some of our internal faculty members. So before I introduce Vera as a scientist, I want to tell you a little bit about why the field of aging research has really taken off tremendously in the last few years and has become such a hot area of research. Really, from the beginning of time, people have been wondering about aging. Why do we age? Why does it happen? How does it happen? Is there anything we can do about it? And aging research has systematically started hundreds of years ago. But for most of that time, it's been more of a descriptive science. We just know what happens as we get old, how pretty much systemically systems break down until ultimately the inevitable happens and the organism dies. But I think we realize now that aging is not an inevitable process of decline, that is just not something we can interfere with in any way. And some of the insight for that came from studies of comparative aging biology, like the research that Vera and her husband, Andrei Zelenov, who's also here, are doing very well. And so things that kind of started this is to see that there are organisms that are very similar in their size and their overall metabolism, that nevertheless vary tremendously in the lifespan that they can display, up to a factor of 10-fold, such as comparing rats to squirrels, or, as you may hear, mice to naked morats. So the conclusion here is that that aging is not just some random, inevitable degenerative process, but something that is genetically controlled, influenced by environmental factors, diet, and a lot of other things. And that opened the possibility to maybe intervene and slow down the aging process so that we might live longer, or more importantly, that we might live more healthy for a longer period of time. And so by studying aging at a molecular and cell biological level, which is what people like Vera and many others in this field have been doing, we gained enough insight into this process to develop rational strategies to interfere with it. And indeed, we now know how to measure aging in an objective way, not just chronologically by counting years, but by assessing how the organism does over time, using DNA methylation maps or looking at the ability of cells to repair damage. And so using these type of technologies, we are now able to assess in relatively practical terms what could slow down aging. That could be dietary interventions, exercise, drugs, and indeed clinical trials are going on right now to see whether this can be translated into something practical for all of us. And so based on that research, aging science has transitioned from like a basic research discipline to something translational, ultimately to the bedside. And so that's a huge potential. Many people are very excited about this. And Vera and Andre have contributed to this significantly. And I think based on that science, we can hope that at some point this old idea of the fountain of youth might transition from a fable to a distinct possibility. So with that preamble, let me tell you a little bit about Vera. I printed out her CV this morning, which is hefty. So she started her scientific education at the University of St. Petersburg in Russia, and then moved to the Weizmann Institute in Israel to get her master's and Ph.D. She then moved to the States in 1999, doing postdocs at McGill University, actually first to Canada, at McGill University and then at Baylor, and arrived in Rochester in 2004 as an assistant professor in the Department of Biology. Now she holds the Doris John Cherry Endowed Professorship of Biology and Medicine and has had quite a run. She's definitely a star in that field, internationally recognized and awarded. She has received a number of awards. I just list a few here: the Cosarelli Prize from the National Academy of Science, the Prince Hitachi Prize in Comparative Oncology from Japan, and our own Davy Award for cancer research, not to forget. He's very well funded, is PI of one program project grant, co-PI of another, multiple other grants, a tremendous publication list, including all the journals that we all like to see: Cell, Science, Nature, Molecular Cell, you name it. The other thing that I find also remarkable is her CV includes, I think, 3 or 4 pages of references that are in the public domain. So these are journal articles or newspaper articles about her research that illustrates how the kind of things that are going on in Vera's labs and in other labs in this research field are really affecting how we think about aging, how we think about preventive medicine, and it illustrates the potential impact that we expect from this type of research. So I'm very happy to ask Vera to come to the podium.
Okay, well, thank you very much for the introduction. Let's see if you can hear me. Thank you for the introduction, Dirk, and thank you to George Abrams, to Mark Taubman for having this series. It's really, I think it's really inspiring to actually have it under these conditions. I think it inspires hope that we are kind of getting out of it. Maybe it will be, maybe the next one will be under more normal circumstances. I hope so. So maybe just something memorable and then to put it behind. But it's also really nice to see people in the audience. So thank you for coming for this event. All right, so let's just dive into the story I wanted to share with you. So this is the fountain of youth. This is something we are really trying to find using scientific method. People were trying to find it for many centuries. So maybe that's the desire that's been there from, biblical and pre-biblical times to, enter these healing waters and then come out rejuvenated and healthy, free of any disease. So that's what we are trying. There are different approaches to find the fountain of youth, right? There's different strategies people use. One of them that we apply in our group is what Dirk already mentioned, comparative biology, where we study species that are naturally long-lived. So here we have mammals ranging from, this is a shrew that lives 2 years, and then all the way to bowhead whale that lives 200 years. So it's 100-fold difference in lifespan. Amazing. And if we could find, or we are finding those molecular mechanisms that are responsible and then apply them to human health. So that has really potential for real breakthrough. So there are different species on this spectrum, right? I started from the shortest lived and the longest lived, and we have other exceptional creatures like naked mole rat, which lives, well, 32, it's outdated slightly. Now the record, although it's not published, but from what we know, personal communications, the record is 40 years right now. So this creature is the same size as a mouse, but lives more than 10 times longer. So that is also remarkable. And this is kind of a strategies that we use, right? So we study those molecular mechanisms. And today I will try to cover a couple of them. And once we identify molecular mechanisms responsible for exceptional longevity and disease resistance, then we can make mouse models. And then eventually, once we see if it works in the mouse, if the same strategy can be transferred from the original animal to the model organism, then the next step is to develop pharmacological interventions. So how can we create the same milieu in a human being? And that was already something that would be translatable. So that's kind of the idea, the philosophy that we use. And I will show you some examples today. We'll talk about just briefly about naked mole rat, because I felt, well, if I don't mention naked mole rat, maybe people will be disappointed. And then we will spend most time talking about bowhead whale, which is something new. We haven't published it, so you'll be the first to hear this amazing research that people did in my group. Yes, we're kind of outgrowing the space. Large vivarium also. So let's just start small with the naked mole rat. So that's where we started many years ago when Andre and I arrived here, and we decided, okay, let's start with something that lives for a long time. And we brought naked mole rats to Rochester. In the beginning, we really didn't know where we would begin, because yes, people knew they live for a long time, but nobody knew the mechanism. And we really, you know, I really loved Dirk's introduction. So there was a lot of descriptive science. And then how do you move from descriptive science to mechanism? So that's really a challenging transition to make. So with naked mole rat, we started studying cancer resistance, because these creatures not only live 40 years, but they're resistant to cancer. They're resistant to all age-related conditions, pretty much all. And what we found was that they have very unique extracellular matrix. They overproduce this molecule called hyaluronan, which human tissues also contain, but we have much less of it. It's of somewhat different structure, shorter molecules. And in the naked mole rat, it's very abundant and it has very long molecules. So, okay, we found it helps naked mole rat avoid cancer. And then the next question was, okay, but can we move it to a mouse? Can we move it to human eventually? So this is the work. It was started by very talented graduate student, Shao Tian. And I will mention his name one more time because he did another amazing work here. So he started with naked mole rats, also actually Chris Hines started, but Shao finished. So here he's standing with naked mole rat. So you can appreciate how small this is. And then this project was completed by Jihui Zhang, graduate student who is still in the lab. So what Shao and Jihui did, they took naked mole rat gene for making hyaluronic acid, Has2 synthase, and put it in the mouse, so making transgenic mouse. And I must say the initial effort was actually funded by a pilot project, pilot grant from Wilmot Cancer Center. So this is how we started. And we started studying these mice. So first we looked at cancer. So here we tried to induce tumors with DMB-ATPA, skin carcinogenesis model. And you can see, so these are controls and these are our transgenics with naked mole rat gene. So they are more resistant to cancer. They were also more resistant to spontaneous tumors. So I'm not showing you the graphs, but well, they do get tumors. So we didn't make them completely cancer-proof like naked mole rats, but the frequency was reduced. And then most exciting, they also live longer, not by, not 40 years, but significantly longer. Again, not to go into a lot of details, so this is the work of Jihui. He found out that high molecular weight hyaluronan represses inflammation. And this is now something beyond cancer resistance. So it actually was something that contributes to lower incidence of other age-related conditions, because inflammation is at the root cause of many conditions in aging. So apparently high molecular weight hyaluronan represses inflammation, and it is also has unique effect on stem cells, where it helps activation of stem cells when needed. So these were, these unexpected effects. But, I just wanted briefly to allude to naked morats, so we have more time to talk about bigger creatures. Just before we go there, I want to mention this animal that is kind of, to blame for many troubles of mankind. No, not this particular one, but bats in general. They are extremely good at dealing with inflammation, and we are studying them in our laboratory. We actually started before COVID, but with COVID, it became even more important to study how naturally, these creatures successfully deal with inflammation, and it allows them to tolerate viruses, not only coronaviruses, but they can live with rabies, they can live with Ebola. It doesn't really affect them as much, so they tolerate them, and they don't develop these cytokine storms, and they're also exceptionally long-lived. So there are tiny bats that live 30, 40 years. So this is maybe something for the future. I won't talk too much about them because you see how spooky they are. So let's move to another topic, which is something that lies sort of at the base of the aging process. So we were always interested, how can we target whatever the underlying cause of aging? And damage to DNA has long been considered such a cause, because once DNA is damaged and the mutation occurred, so we cannot really fix it. Our DNA is subjected to lots of insults that come either from external factors or from errors in DNA replication and transcription. So over the course of our lives, our DNA is bombarded with all of those events. And what eventually happens, and actually that's kind of a question people were asking, and those who criticize this theory that, well, DNA repair is so important, was saying, well, but we do accumulate mutations with age, so that's no question. But can those mutations really explain aging? Because, well, there is a lot of non-coding DNA in our genome, and many mutations don't really hit important genes. But a new thinking that started to emerge in the past several years has been that it's not only the mutations or change to DNA sequence, but it actually is a change to the epigenetic structure. And this is something that is much more profound, and mutations or rearrangements can affect many nearby genes just because of changing this DNA packaging. So there is this concept of epigenomic drift. So this drift occurs, and this is something that can really affect the process. So in other words, here is our young cell. DNA is neatly packaged into specific domains, and genes are turned on and off when necessary. And then epigenetic landscape looks like that. Whatever has to be active is active, whatever has to be repressed is repressed. But over time, because DNA goes through these rounds of insults and repair, so eventually it's like when you take your sock drawer and take things out, put back. I mean, it's hard to put it as neatly as it was there initially. So in the end, it becomes a mess. And this is what epigenetic landscape becomes with age. It becomes smoothened. So this is something, the theory, I mean, it's been like a hypothesis, and I would like to show you, what evidence we get for that, and then what do we do about it? How do we bring this landscape back into this young, youthful shape? So let's start with different enzymes that play a role in shaping epigenetic landscape. So there are many epigenetic modifiers, and one of them we've been studying in our group for a long time. It's called sirtuin 6. It's been kind of notorious in the aging field because it's a homologue of yeast sirtuin gene that was shown to extend lifespan in yeast. And what we demonstrated already 10 years ago, that it's also important for repair of DNA double-strand breaks. It gets recruited to damaged sites, and it helps sort of prepare the platform for other proteins to come and repair. And then another function of this gene is to maintain chromatin. So it kind of does both of these. It keeps heterochromatin together, and it represses repetitive elements which are found near telomeres, centromeres, and also other parasitic line 1 and signs and different transposons that our genome is riddled with. So SIRT6 helps keep all of these elements in check. So that's the function. And I should also mention that it was shown that if you knock it out in mice, they age prematurely, and if you overexpress it, they live longer. So here is a nice connection. But what we wanted to do is to kind of broaden it and say, okay, but can we see correlation between better handling of double-strand breaks and longer lifespan? So here we use this collection of rodent species. We have like 20 of them, or actually more than 20 now. We have primary cells, primary tissues from all of these species that live, anywhere from 3 years in a mouse to 40 years in the naked morat. And we measured the efficiency of double-strand break repair. What we found, again, I'm going very fast to show you some new data. We found a very strong positive correlation between better DNA repair and longer lifespan. And then when we started looking for the mechanism behind, we found that SIRT6, actually, difference in SIRT6 activity was responsible for a major portion of this correlation. And for example, SIRT6 in the mouse was a weak enzyme, but SIRT6 in the beaver was much stronger. And we could kind of swap them, vice versa. We even, teamed up with Dirk Bowman, and he expressed these different versions of SIRT6 in Drosophila and showed that with beaver SIRT6, flies were living longer than with mouse SIRT6. So that was really like the last proof we needed to get it published in Cell. So yeah, that was a really nice story. But then what we showed, that across species, double-strand break repair is very important. Now, what about within species? What about now, these are all long-lived animals like beavers, naked morats, but what about humans? Will more active SIRT6 benefit people for longer, human longevity, and also are long-lived human individuals, do they have more active SIRT6? So here, just for inspiration, I'm showing you some of the American centenarians. So actually, Mr. Image lived in New York City to the age of 111. He's been very active. He was working at his office, I think, till age 100. So that's something we wish for everyone. And this lady lived to a ripe age of 119. She's been also exceptionally healthy. So what about these people? How do they do it, and whether this better ability to repair DNA damage has anything to do with it? So this is the work of a postdoc in our group, Matt Simon, in collaboration with Yusin Su from Columbia University. So what Yusin did, she sequenced cohorts of human centenarians, actually from New York State, and she was looking for any mutations that are overrepresented amongst human centenarians. And you often find many mutations, but most of them, or I wouldn't call them mutations, sort of polymorphisms, most of them are in non-coding regions of the genome. So it's really hard to tell what they do. But she did find 2 coding SNPs in human SIRT6. So these were overrepresented among Ashkenazi Jewish centenarian cohort compared to controls. So controls would be Ashkenazi Jewish individuals with no history of exceptional longevity. These 2 polymorphisms were in the C-terminus of SIRT6, which is, by the way, the same region where we found important differences between mouse and beaver protein. And then what Matt Simon did, he engineered these into, a vector and, to make recombinant protein. And then we analyzed its functionality in DNA repair assays. So here we are looking at the DNA repair assays comparing wild type human SIRT6, so like SIRT6 in any one of us in this room, compared to centenarian version. And we see that it is better at repairing DNA breaks by homologous recombination or non-homologous end joining. Not a huge difference, but again, centenarians don't live 3 times longer than the rest of us, just some percentage longer. So we found significant difference. We also looked at spontaneous DNA damage in cells that were harboring either wild type version or centenarian version. And again, there were fewer DNA damage sites in cells with centenarian version of SIRT6. And centenarian SIRT6 was also better at repressing those line 1 elements. I mentioned the genomic parasites. And this is expression of line 1 in the presence of wild type SIRT6 versus centenarian version. And these two, I didn't go into details, but these are those individual alleles. But in centenarians, they were always in linkage disequilibrium. So we were testing one individual and both together to make the centenarian. Okay, so from that, we were quite interested to find out biochemically what's different about centenarian protein. SIRT6 has 2 biochemical activities. It can deacetylate proteins, and some of its favorite substrates are histones, so it deacetylates them, and this is how it enforces heterochromatin. But it can also ribosylate proteins, so it can transfer ribose on the protein. So we measured each of these activities in vitro. So here we are looking at deacetylation, and when the signal disappears, it means SIRT6 deacetylated the substrate, in this case H3K9. And to our surprise, centenarian protein was less active. So John Gigas is sitting here. I think he was the first one to do this assay. He's MD PhD student in the lab, and at first we just couldn't believe our eyes. But it was confirmed since then by several people in the group. So indeed, centenarian SIRT6 is less active as a deacetylase. But it has also the second activity, mono-ADP ribosylase. And here we saw that centenarian SIRT6 is much more active than the wild type. So very interestingly here, we got this kind of separation of function that in centenarian protein, we have one function reduced and the other one is enhanced. So we were also quite curious about interacting partners of SIRT6. Are they affected by the centenarian variant? So this is the work done by Greg Tomblin. He's our guru of proteomics. So he did IP and mass spectrometry to look at interacting partners of SIRT6, wild type versus centenarian. We didn't find any new interactions, so which means, well, centenarian SIRT6 still does the same thing. But we found a group of proteins that were binding stronger to the centenarian version. And the best hit here with most confidence was lamin A. And this is a very fascinating protein. So lamin A, mutations in lamin A are associated with many human syndromes. The most known of them is Hutchinson-Gilford progeria syndrome, which is a very severe human premature aging syndrome. So you've probably seen these children. They usually survive, maybe just to early teenage years, die mostly of stroke, cardiovascular disease, but they also show multiple features of premature aging. So what lamin A does, it provides this structure to the nucleus. Lamin, it's kind of a scaffold that maintains the structure within the nucleus. And now you see how relevant that is to this epigenome idea. So if this structure gets messed up, you end up with a premature aging syndrome. Another interesting fact about lamin A, that not only it's associated with disease, but polymorphism of lamin A were also found to be overrepresented among human centenarians. So you could also have it work better. And here what we showed was that centenarian version of SIRT6 binds more strongly to lamin A. And that perhaps promotes better maintenance of chromatin structure in the nucleus. So now let's switch gears a little bit and talk about bowhead whale. So this is a very exciting animal. Not only it lives 211 years, or probably more than that, 250. It's really difficult to, to get accurate numbers on these lifespan of these animals. It's also extremely large, which makes it, also a very interesting model for cancer research studies, because you imagine if the body is that large and their cells are about the same size as human cells, but then they should experience much greater statistical risk of developing tumors, but they don't. So they're not only long-lived, but they're also exceptionally good at, preventing cancer. So we were interested in bowhead whales for a long time. The work I'm going to present you was done by postdoc in the lab, Denis Fersanov, and graduate student Max Zucker. So here they are standing in Barrow, Alaska, between the two jaw bones of bowhead whales. So you can appreciate how large this animal is. So they traveled to Alaska to collect primary cells from whales, which was not an easy task. In Barrow, Alaska, there is no laboratory, with sterile tissue culture hoods. There is not even FedEx service. So they actually had to take those samples and then fly back and bring them in their checked luggage. So that was tough, but they succeeded to collect samples from several bowhead whales and brought them in the lab. And now we've been analyzing them. So, to the question of cancer resistance. What I presented to you, this puzzle of how come large animals don't get more cancer, is also called Petus paradox. And it's been studied before in elephants, because elephants are large. Of course, compared to whales, they're nothing special, tiny creature. But they're quite large compared to other terrestrial animals. And what was found for elephants is that they have multiple copies of p53 tumor suppressor genes, which perhaps gives them a higher propensity to undergo apoptosis or programmed cell death. And that was proposed as a mechanism of cancer resistance in the elephant. Although I would say that that's probably a very wasteful strategy. If you're killing a lot of cells, then, not before long, you will run out of stem cells. It will be a problem. So we still don't know how elephants deal with it. But the question we asked first was whether whales do the same thing. So we looked at p53 activity, comparing whale to human. And we didn't see anything remarkable. If anything, p53 was even less active in the whale than in human. So then we thought, and also from genomic analysis, it didn't seem like whale has any obvious amplifications of tumor suppressor genes. So then we asked this basic question. So how many mutations, how many hits does it take to make a tumor out of a whale cell? And this is from classical Bob Weinberg studies. We know that for human fibroblasts, it takes 5 hits. For mouse fibroblasts, it takes 2 hits. You can just mutate either RB or p53, activate RAS, and you've got a tumor. In human, it's not so easy. You need to mutate both p53RB, also pp2A phosphatase, activate RAS, activate telomerase. So what about whale? So first we tested whether you need to activate telomerase. We cultured whale fibroblasts and we observed them enter replicative senescence, which suggests lack of active telomerase activity, also confirmed by trap assays. So we see that we do need to activate telomerase. Otherwise, whale cells will enter senescence. So senescence has also been, really something quite a fanciful word lately. So people have been looking at senescence as one of the driving causes of aging through induction of inflammation and so-called SASPs. Senescence associates secretory phenotype when cells secrete all these inflammatory cytokines and promote inflammation. So we compared SASP between whale and human. And you can see, so here is one example, IL-6. Highly induced in senescent human cells, but not so much in whale. And that's true for many other cytokines. So SASP phenotype is really much milder in the whale. So you see it's shifted towards humans. So this may be, one useful thing for whales. They are more resistant to this inflammation induced by senescent cells. But now back to our question of oncogenic hits. So we started introducing those hits into whale cells and then injecting them into mice and looking whether they form tumors. So we mutated p53, introduced telomerase, activated RAS, no tumors. So then we also inactivated RB, still no tumors. We could see small colonies growing in soft agar, but no tumors in the mice. So now we made four mutations, activate telomerase, inactivate RB53, and mutate RAS. And we see tumors forming. So we were quite shocked because we expected whales should need many more hits than human. But here we have only 4 hits instead of human 5, and they're already forming tumors. If we also mutate PP2A phosphatase, we get even larger tumors, but even without PP2A mutation, it was enough for the tumors to form. So that was the conclusion. Only 4 hits are required for malignant transformation of human fibroblasts. And then that made us thinking, so our initial assumptions that there will be more tumor suppressors is wrong. But still whales don't get cancer, at least not at high frequency. So they must be doing something different. And then we thought, well, but all these tumor suppressors we've been talking about, at least, p53, RB, PP2A. So these are tumor suppressors that sort of are gatekeepers. They kill cells that accumulated mutations. But maybe whale DNA repair or genome maintenance is so good, they never even accumulate those initial mutations, so they don't need to kill their cells. So we thought, okay, let's test this possibility. We started looking at DNA repair in the whale. We measured base excision repair, nucleotide excision repair compared to human cells. We didn't see big difference. And then we got to double strand break repair. So that's the same pathway that we showed to correlate with aging across multiple rodent species. And we saw that bowhead whale is way better than human at non-homologous end joining. And not only are they more efficient, but they're also more accurate. So if we were measuring, error proneness of non-homologous end joining in accurate events, they were much higher in human than in whale. So this is percentage of accurate joining events, just 15% in human, 60% in whale. So they are really good at it. And then we've done various other assays. So here we also added mouse and cow as the closest relative of the whale that doesn't live in the water. And you can see, so this red, so these are larger deletions, smaller deletions. So mouse makes a lot of deletions when repairing double strand breaks. Cow is a little bit better, human better, but bowhead whale, like, come on. I mean, it doesn't, almost no deletion events, mostly precise events. So we started thinking, okay, so what's the mechanism for this accurate and efficient non-homologous end joining? And we looked at various proteins that are involved in these pathways, compare them human to whale. Some of them were actually even lower in the whale than in human, like Q80, Q70, they were lower. But 2 proteins really stood out. PARP1 was present at much higher level in the whale, and KIRP. So this protein was really like off scale in the whale. So what is KIRP? We really didn't think much about this protein before we started working with whales. So the name stands for cold-induced RNA binding protein. And as the name suggests, it's induced, expression is induced by cold exposure, and then it binds RNA and kind of changes transcription pattern to adapt to cold. But it has another role, and there is only one paper really talking about it, that KIRP is also involved in double strand break repair. So it's kind of moonlighting double strand break repair. It is recruited to double strand break, and then it binds to to PARP1 and those PARP moieties, and also helps recruit other DNA repair factors, which kind of resembles what SIRT6 does. So they're kind of working together at the same interface. So then we thought, okay, so KIRP may be doing it, but what if we take now bowhead whale KIRP and introduce it into human cells? Will it improve DNA repair? So here we have now human fibroblasts expressing bowhead whale KIRP And this is non-homologous unjoining efficiency, this homologous recombination efficiency. You see, it can improve DNA repair in human cells. I mean, that was super exciting. So we just take this protein, move it. So then, of course, we ask the question, but can we just improve DNA repair by inducing our endogenous KIRP, just by brief cold exposure? And yes, we can. So when cells were briefly exposed to 22 degrees instead of 37 in the incubator, efficiency of DNA repair went up. So that is kind of amazing because we know whales, they live in, bowhead whales live in Arctic Ocean, plus 4 degrees Celsius, and this is why perhaps they evolve these large quantities of KIRP. From traditional medicine, there are always these ideas that you have to jump in cold water, brief exposure, it somehow stimulates your immune system. I was somewhat, suspicious of these claims until we got this result. So maybe indeed there is some rationale in it. So to recap what we found in bowhead whale, attenuated SASP, which suggests lower inflammatory responses. And then they have more accurate and efficient double-strand break repair, which is mediated by very high expression of cold-induced RNA binding protein, which perhaps promotes not only better genome stability, but maybe also epigenome stability. This is something that we are testing right now. So again, another strategy that we can borrow from whales. So perhaps, if we find ways to induce our endogenous KIRP, by cold exposure, brief one, or by some other pharmacological means, so that may actually improve our genome maintenance. And let me very briefly mention another project that is now rapidly sort of gaining momentum in the lab. So the omics of longevity. So this is the work of Yu-Yang Lu, very talented postdoc bioinformatician, and Greg Tomblin, again, our proteomics expert. So we took all of these species that I was talking about, collected tissues from them, extracted RNA proteins, and now we are doing comparative omics to find now on this more global and biased scale if there are specific signatures of longer life. And what I can share with you, so these are gene pathways that Yu-Yang found to be either negatively in blue or positively correlated with longer lifespan. So what really stands out here? So these are the positively correlated pathways: DNA repair, homologous recombination, double-strand break repair. So here we took completely unbiased approach and we got to the same conclusion. So then, what Yu-Yang did, and this is like completely amazing, he took all these signatures and then he did regulatory network analysis on it to find what kind of transcriptional regulators control these genes differentially. So this is kind of a functional genomics approach to the comparative biology data, which hasn't been done before. So what he found that genes that are negatively associated with lifespan are controlled by circadian transcription factors, which perhaps suggests that these genes, and these are genes that are typically involved in metabolism, so they kind of promote those metabolic processes that shorten our life. So they are under circadian control to limit their expression so that we are not always high, but they're only high as long as it's needed. And then in longer lived species, they are somewhat lower. And now what about these most exciting positively correlated genes? So these are under control of pluripotency factors. So this was like another really amazing finding because recently another topic that's been emerging is epigenetic reprogramming in aging. So from the time when Senior Yamanaka showed you can reprogram somatic cells into iPS cell, that was more like, people were thinking, okay, we can make different stem cells. But then it was followed up by several studies where those factors, pluripotency factors, were expressed in the mouse, which resulted in rejuvenation. And I was thinking about it more like, well, this is a really kind of unnatural twist. You turn, the clock back by sort of de-differentiating the cell. But now, based on Yu Yang's work, we see that actually this is the same strategy that's used by evolution to adjust lifespan, because in longer-lived species, the downstream targets of these pluripotency factors are expressed at higher levels, which probably helps them maintain sort of regenerate tissues better during their long lifespan. So here, this is the landscape I showed you. So how do we sort of reverse the clock from here to here? So we tried one thing, I told you about CERT6, that we suspected, that it's associated with lifespan across species. But here we just took fibroblasts from older people, just isolated, fresh isolates, people, aged 60 to 80 years of age. And we expressed CERT6 for 2 weeks. And then we measured methylation age of the sample. So this was in collaboration with Steve Horvath's, who developed this Horvath's clock. So now we have this amazing biomarker of aging. And what we see, so we had 10 cell lines from 10 people of older age. Epigenetic age went down. All of them except one. So just expressing CERT6 for 2 weeks was sufficient to lower epigenetic age. So this is like, so exciting, because now, okay, if we find pharmacological activator of CERT6, I mean, maybe we could turn back the clock. So, there are several activators reported in the literature, but most people were looking at activators of deacetylation just because it was easier. But now, based on our work on centenarian CERT6, we decided, well, we need to find activators of mono-ADP ribosylation, because we know this is the activity that's associated with human longevity. So we tested known activators of deacetylation for the ability to activate mono-ADP ribosylation. And many of them really did nothing. But there was one that actually activated both deacetylation and ribosylation. And this is a this is a molecule called fucoidin, which, is not exactly what we wanted to find. It's not a small molecule. It's a short sugar polysaccharide. And it is found in seaweed. And as we all know, seaweed is very, big part of diet in Japan and South Korea. And these are two countries with the highest life expectancy. They eat a lot of seaweed. And, again, we really don't know if this is because of CERT6 activation, but I always say, if you want to have a take-home message, eat some seaweed, buy some sushi, definitely not going to hurt anyone, but maybe it will activate your CERT6 as well. So let me then, come back to this picture of epigenetic drift. How can we reverse it? So now we can offer different strategies. Right, so to achieve epigenetic rejuvenation, well, Yamanaka factors, that's proof of principle was shown in mice, but there are many dangers associated with this forced de-differentiation. You can get cancer, that's what happens. So ideally, we would want to find a much safer strategy, and CERT6 may provide it because, well, it doesn't change the identity of the cell, but it can help repackage chromatin to this, more youthful landscape. We discussed KIRP from Bowhead whale, again, more research is needed, but maybe that's another strategy. And there may be many other enzymes and epigenetic activators that, yet to be discovered or adopted for this purpose. So I think, what I would like to conclude with is that, yes, we actually getting closer and closer to developing strategies to actually reverse aging and to go beyond treating individual diseases, but to actually reverse aging process as a whole and increase human health span and lifespan. So I would like to thank members of my group. I was naming them as we were going along. So here you can see Matt Simon, Greg Tomblin, Dennis is hiding here, so he's Bowhead whale researcher, Shao Tian, before he left our group to move on. Of course, I would also, Max Zucker is standing behind here, Jihui Zhang. I would like to thank my lifetime collaborator, Andrei Siluanov, who really co-directed all of these studies together with me. And we have many outside collaborators, these project grants and now these big consortia looking at omics of aging. So many people contributed to this work. And, I was, it was really my, honor and pleasure to be here at Rochester. And it's been, extremely amazing environment for all these studies to flourish. And I would to thank all my collaborators here and for being supportive and also being so open-minded for allowing us to pursue these crazy projects. And we are looking forward to many more discoveries. So thank you very much. Yeah, well, let me go back to this. Yeah, well.
Can you repeat the question for the people at home?
Okay, so the question is whether these two amino acid changes are sufficient to result, this change in activity. Well, so we have, pretty, big changes, I would say, from polar amino acid to charge, from hydrophobic to polar. So it's not completely identical. What I can also say that this C-terminus of CERT6, so the catalytic domain is in blue and yellow is the C-terminus. So it was excluded from the crystal structure because it's very flexible. It's unstructured C-terminus. We don't know exactly how, but it's unstructured and it really regulates the activity of the catalytic core. So I would think that even small changes within this C-terminus can confer, kind of tune the protein differently. And now your second question was about KIRP. This is, you really have very sharp eye. I kind of skipped over that for the sake of time. So thank you for bringing it up. Yes, because we can really talk about this amazing fact. So KIRP is highly conserved. So there are only, I think, 5 amino acid differences between human and whale KIRP. Actually, Dennis tried to mutate each one of them to see, and we do see a difference. But also what we see that there is a difference in the sort of codon usage between human and whale KIRP. So when Dennis tried to overexpress human KIRP in human, he couldn't get these high levels easily as in with the whale because some of the codons are kind of non-optimal. So in the whale, also the codon usage was optimized to allow this very high expression. So maybe the difference is not so much, these 5 amino acids, but also the mRNA, the protein levels and the codon usage. So of course, this raises a question, can we, it's a bit more difficult than for human to upregulate KIRP to the same level as in the whale. So maybe we have to also look into some pharmacological ways of doing it, not just jumping in cold water. So, still there is more research to be done on that. So thank you for this question.
So maybe the health advantage of going to the sauna is not the sweating, but the jumping.
Yes.
Jim.
Well, this is a very rare variant. Oh, the question was how common are the polymorphisms that I was showing in CERT6. This is a very rare variant. And we kind of struggled with it because it's really difficult to get to statistical significance levels that are needed with the, population. We have like, 600 centenarians, and to have really solid p-values, we need about 10,000. So something else we did, we just took entire database of human genomes in the GNOME AD database, not on centenarians, but everyone. And we see that this polymorphism is also overrepresented in people older than 75. So I think, that's kind of reinforced our belief that it is indeed associated with longevity, because on centenarian data alone, those rare variants, are very difficult to validate.
Do you have any sense that there's a difference in terms of how these animals respond to what state of stress?
Yes. Dennis spent probably a year looking into oxidative stress response in the whale. And we see that, whale is more resistant. And also it has a lower sort of background ROS levels. So it probably, manages its mitochondria better. Although here there is a caveat that larger species tend to have somewhat slower metabolism and somewhat fewer mitochondria. So that may explain why just normally they have less ROS, but in the end, it may contribute to less DNA damage as well.
If there are questions from people on Zoom, you can put them in a text and we will put them to Vera. Can you maybe? I wonder if changes mRNA translation that may contribute to longevity, because it is known the inhibition of translation for long lifespan in yeast.
Yeah, that's a very interesting possibility. Yeah, we have a strong RNA group here, so I definitely, would be happy to get any feedback on how, because KIRP, we kind of focused on its moonlighting function in DNA repair, but it may also very well be regulating RNA sort of metabolism in the cell and optimizing it. So definitely, that's a very good point, something else for us to study. Oh. Yeah. So the question is whether, because, well, there is a known fact that women live longer than men, and also females of some species, not all of them. Mice, for example, it's not true, but there are many species where females live longer. Whether this can be explained by high levels, of pluripotency factor control genes. To be honest with you, I don't have an answer, but this is a very interesting possibility. And, I will email you, Yang, as we finish to test that, because I think that's a fairly straightforward question to answer. I don't think anyone really looked into that.
To follow on Mark's question about reactive stress, do you have a sense of DNA or repair? How they weigh in on the aging process? Damaging DNA get repaired. Damage to repair.
Well, yeah. So, there is this theory of a vicious cycle. Like there is more DNA damage, then it damages the repair systems, and then like everything, goes down the drain, basically. So I think that takes place during aging, because for many organisms, humans included, like we maintain our physiological capacity till middle age sometime, but then, in our 50s, things really start to deteriorate rapidly, and maybe the process you mentioned contributes to that. So I think preventing, DNA damage and epigenetic deterioration should we should be able to reverse or stop this vicious cycle. But again, you know, to the balance, it's difficult to really say because aging is a very multifaceted process. So many things go bad, and to say, okay, which one is the worst? Of course, every person studying a particular process would say, okay, you know, my process is more important. I guess the real test would be, you know, how can we reverse it? Whatever manipulations will be able to reverse all of them, because, okay, we can, you know, improve proteostasis a little bit, we can improve DNA repair a little bit, we can, you know, give antioxidants, but so far, all of these treatments were only giving very incremental improvement. So now what we are looking for, you know, with these epigenetic reprogramming strategies, maybe we can really improve, make a dramatic improvement. We don't know yet, but that's where we're trying to get with this.
Okay. One more question.
Have you seen if there is a difference in the activity of certain sex depending on the age of the animal? Does it mean there is a time where they, you know, it stops decreasing, or when it stops to being able to be activated?
Yeah, so that's a very good question. Yes, we looked into that. You know, just the level of CERT6 doesn't decline very much. So it's not, you know, sometimes for many proteins, people want to show it really drops down with age. No, we didn't see it for CERT6. But the activity may decline because what happens as we get older, and I mentioned these two functions of CERT6, DNA repair, and also maintaining all those repetitive elements and telomeres, centromeres. So we need it at those repetitive elements, but then there is more damage, DNA damage, so it gets relocalized. So this is so-called this redistribution theory. And when it gets relocalized, there is less left to attend to chromatin jobs. And here what happens, transposons start to get expressed, they trigger inflammation. So yes, it does take place. So there is reduction in activity over time. Yeah, hope that answered your question.
Good. Great. Thank you very much, Vera.